What I plan to do is really give you an overview of the research that I've been carrying out in the last 10 to 15 years, predominantly in the area of herbicide resistance. So just to start out by putting things in context, this is a slide that I guess most weed scientists take pleasure in putting up at the beginning of their talks. This was a study carried out by ERCA in 2006, just looking globally at the potential and actual yield losses in all crops globally from pathogens, pests and weeds. And what it's shown is that the actual potential loss from weeds is about 35% compared to about, I think it's 18 and 16% for pests and pathogens respectively. So potentially, weeds can cause far greater yield losses than both pests and pathogens. But actually, when you look at the actual average losses globally, it's about equal between the different groups. And that's largely the sort of success in reducing that potential loss to a relatively manageable actual loss has largely been as a result of the use of herbicides. And herbicides account for 46% of the global pesticide market. Now, of course, one of the unwanted consequences of that use, that widespread reliance on herbicides for weed control, has been the evolution of herbicide resistance. And this graph just summarises, as it's a sort of chronological summary of cases of herbicide resistance. So herbicides were first introduced shortly after the Second World War. The first cases of resistance were reported in the triazine herbicides. The first case was in 1968. And as you can see, for the triazines, there was a fairly rapid increase in cases of resistance through the 1970s and 80s. Now, this was a sort of, there was research done on triazine resistance from the point of view of biological interest and understanding how resistance was evolving. But triazine resistance never really became a major agronomic issue. And that was really because there were a number of other modes of action. The 70s and 80s was really a sort of a period where a number of new herbicide modes of action were discovered. So triazine resistance never really became a major agronomic issue because there were always other herbicides which could be used. The other group I'm focusing on there are the ALS inhibitors, probably the most widely used herbicides globally, and those were introduced in around about the late 70s and early 1980s, and we saw a very rapid occurrence of resistance to ALS inhibitors. Now, about 100 weed species globally with documented resistance to ALS inhibitors. The ACCA's inhibitors are grass selective herbicides, similarly introduced. Most of those active ingredients were introduced in the 70s and 80s, and again we saw a fairly rapid evolution of resistance. And the final group I just wanted to focus on here are the glycines. Now, there's really only one glycine herbicide, that's glyphosate, probably all familiar with the name Roundup. And again, you might even argue that ALS resistance and ACCA's resistance never became a huge issue because around about the time those two resistances were becoming very severe, we saw, certainly in North America, we saw the introduction of Roundup Ready GM herbicide-resistant crops. But unsurprisingly, and I'll talk a bit further about this as I get into the talk, the introduction and widespread adoption of those Roundup Ready crops has resulted in the widespread evolution of resistance to glyphosate. So really, unsurprisingly, herbicide resistance is a global phenomenon. And there are really very few herbicide modes of action which aren't prone to the evolution of herbicide resistance. And this slide just gives a sort of global picture of herbicide resistance. The darker the shading, the more species that have been documented in those countries with resistance. And unsurprisingly, currently we see the most severe cases of resistance in North America, Australia and Western Europe, where those herbicides have been used most widely and for the longest period. But also we're increasingly seeing a big resistance problem in South America, China, India, as more and more herbicides are being used in those countries. So this just basically summarises some of the worst offenders. This chart just lists a number of weed species by the number of modes, sites of action for which herbicide resistance has been documented. So I'm going to talk a little bit about Lolium rigidum. I worked for a number of years in Australia, and that's the #1 weed problem in Australia. I've underlined 3 major herbicide-resistant weed problems that we have in the UK. So that's with Alopecuris myoceroides, blackgrass, Lolium multiflorum, annual ryegrass, and wild oats of Vena fatua. So just to give a brief overview of what we know about the mechanisms of herbicide resistance, we basically know for herbicides to work, they have to penetrate the plant, they don't have to be translocated to the active site, to the target protein. They have to accumulate at that target protein. They then bind to the protein and inhibit the enzyme activity. And we've really documented, to varying degrees, resistance evolving through interference at all of those stages. Now we can basically distinguish between target site resistance and non-target site resistance. And what I'm going to talk about mostly today, and my major interest, is in non-target site resistance. So target site resistance is typically the result of single nucleotide polymorphisms which lead to an amino acid change in the target protein, which means the herbicide is no longer able to bind and inhibit that protein. It's typically inherited in a Mendelian fashion, so these are major single genes. And it leads to what I would term specialist resistance. So target site resistance gives you resistance to a single mode of action. All other, because it's target specific, all other herbicide modes of action will continue to work. Non-target site resistance is less, we know less about it, and as I say, that's a major focus of our research. It's quantitatively inherited, and it typically gives rise to a much more generalist type of resistance. So evolution of this non-target site resistance, which is obviously often a type of upregulation of metabolic pathways which detoxify herbicides, can give rise to a very broad spectrum of resistance. So often selection with one mode of action leading to evolution of non-target site resistance can actually confer resistance to a whole host of other herbicides to which the population's never been exposed. So potentially a much bigger problem. So I just wanted to sort of, before getting into the body of the talk, just sort of present a model for how I think or how we approach herbicide resistance. So there are really 3 areas on the left there. There's the idea of we need to understand the fundamental mechanisms of resistance, so the way in which plants are able to evolve herbicide resistance, or the mutations and biochemical mechanisms which confer resistance. Probably my greatest interest, and where I've focused a lot of attention, is on the sort of ultimate causation, or the what ecological and evolutionary processes lead to the selection and the spread of those mutations through populations, and then through that understanding, trying to develop management strategies which can prevent or delay the evolution of herbicide resistance. And what I would say, my sort of, through the 15 years that I've worked within herbicide resistance, this is a personal view, I guess, but what I've come to realise is that there's a lot of work, a lot of very good work done to establish the mechanisms of resistance. So we've become very good at establishing the molecular mechanisms and documenting and describing resistance. I think where we still have to make further progress is really understanding some of the ecological and evolutionary questions about how resistance has evolved, and using that knowledge to develop, I sometimes use this term evolution proof, which is probably stretching it a bit far, but developing management strategies which are sort of resilient to the evolution of resistance. So my final sort of introductory slide, the way I've sort of thought and the way we think about herbicide resistance, and everything we do is focused on management. But really, there's sort of an emerging, this field of applied evolutionary biology seems to be quite active at the moment, and the recognition that not just in agriculture, but in healthcare and throughout, and also when we think about conservation and environmental questions, human activity is really driving rapid rates of evolution in organisms, in agriculture, healthcare, and in the wider environment. So some of the questions that I've been interested in answering during the time that I've been working in herbicide in this area is to understand the genetic architecture of resistance to herbicides, looking at costs of resistance, so understanding if the evolution of herbicide resistance is associated with trade-offs and whether we may be able to use a knowledge of those trade-offs to design management strategies which select against resistance when the herbicides are not used. A big question is given a diverse armoury of herbicides, what is the best way to employ that? Is it through rotations of different modes of action? Is it through sequences of different modes of action? Or should we use mixtures? So that's a question we've looked at in some detail. And then finally, another important question, just really understanding how important is the movement of resistance genes across the landscape. So is resistance evolving frequently and independently on different farms? Or are we getting few evolutionary events and then the spread of resistance across the landscape? So, as I say, the way I've been thinking, or when you think about these important questions, which are all important for resistance management, they also cut to the core of some quite fundamental questions in evolutionary biology. So, the importance of novel mutations versus standing genetic variation, minor and major genes, regulatory and structural genes. And we're becoming increasingly interested, I'm not going to talk too much about it today, but we're becoming increasingly interested in recent research in looking at whether there's an epigenetic component to the evolution of herbicide resistance. Costs of resistance, so again, fundamental questions in evolutionary biology about trade-offs and whether evolution of novel traits is associated with costs or with trade-offs. As I say, we're interested in how by sort of creating more heterogeneous application of herbicides, what impact that has on the evolution of resistance. And particularly, I talked about these two modes of resistance evolution, which I called specialist and generalist, the target site and the non-target site, whether those different strategies preferentially select for different types of resistance. And then also, as I say, we're interested in sort of some of the evolutionary dynamics and whether resistance evolves rarely and spreads, or whether actually resistance is evolving independently on many farms. And that has important consequences for how we think about managing herbicide resistance. So I'm going to, for the next few slides, I'm going to talk specifically about the evolution of resistance to post-emergence herbicides. So these are herbicides which are used in the crop. Much of my talk does focus on arable systems. The two particular groups which I've already introduced are the ACCAs and ALS herbicides, and I'm going to use two case studies here. I'm going to go back a few years and talk about some of the work I did in Australia, and then move to the sort of current situation and talk about what we're doing to look at the blackgrass problem in the UK. So just to give a bit of context, as I say, I'm going to start out talking about, I spent 6 years as a postdoc based in Perth in Western Australia, just over here. So the green shaded area shows you the wheat growing region in Australia. It's characterised by very low rainfall, low yields, so average yields 1.5 tonnes per hectare compared to sort of about an average of about 8 tonnes per hectare in the UK. Very large farm sizes. Very little light, dry, sandy soils, so very little soil cultivation. And that means for weed management, the whole system is heavily reliant on herbicides. And often, and I'll return to this issue, often because of large farm sizes, because of low margins, also often with farmers using lower than recommended herbicide rates. And finally, this species, Lolium rigidum. Now, Lolium is an annual ryegrass species. It was introduced to Australia as a pasture species, and it's now far and away the major resistance problem in Australia. When I started work in Australia in about the year 2000, a survey showed that about 40% of randomly sampled ryegrass populations were resistant to both ALS and ACCA's herbicides, and those were the two major herbicides available for ryegrass control in wheat crops. Oops. So throughout my work, we quite often use modelling approaches to integrate the knowledge. We use empirical and sort of theoretical modelling approaches to really integrate the knowledge we're gathering into models to then try and design novel management strategies. So from early on in Australia, one of the key questions we were interested in answering, and we developed models to address this question, is given two effective herbicide modes of action, what's the best way to deploy those herbicides? So basically the options might be to use one until it fails and then use the second one, to use them in rotation, so 1 year of one followed by just rotating them on an annual basis, or using both in a mixture in each year. And we did some modelling studies to establish what might be the best strategy. Now if I did this again, I might present it somewhat differently, but this is from a publication back in 2003. On the x-axis here, we've got population size. So this is basically asking the question, under a set of conditions, we know that a bigger the population, the more likely resistance is to evolve. So this is basically saying, under a given set of conditions, using these three strategies, how big would a population have to be for resistance to evolve? So basically, it's comparing the risk of resistance under these different strategies. So in this instance, we're just looking, under what situations resistance doesn't evolve. And what we find is we need the mixture strategy is robust for much larger populations than both the rotation and the sequence. And we don't see any, in our models, we didn't see any advantage of the rotation strategy over the sequence strategy. And this is just sort of looking at the flip side of that, looking under what conditions resistance to both of those herbicides evolved. And again, the mixture strategy comes out as the best strategy. Now, of course, there's an environmental angle to this. It's much better to use. We're talking about using twice as much pesticide to control this weed. There's also an economic context. But just taking those things aside, we show that mixture strategies are effective for delaying and in some cases preventing evolution of resistance. And I think importantly, we found that rotation does not delay evolution of resistance. And I think there are interesting parallels, and it's quite encouraging to see. There was a recent workshop in the US bringing together people working in pesticide resistance to anti-cancer drugs, and antiretrovirals, antibiotics. And this sort of really this concept of using mixtures is similar to the idea of combination therapy used in healthcare. So mixtures work. They can delay evolution of resistance. What I should say about the simulations we carried out here is we were making the assumption this is just only looking at target site resistance. So this is only concentrated on the evolution of those target site mechanisms. So we expanded the context of what we were doing to incorporate non-target site resistance. We made some fairly simplifying assumptions that this metabolic cross-resistance was a single gene trait, which I think we know subsequently and even at the time probably isn't strictly true. But we just wanted to take a fairly simplistic approach to start to consider what impacts non-target site resistance might have on evolution of resistance to those two modes of action. And the question that underpinned that really was, could herbicide rotation result in the rapid evolution of metabolic resistance? Were we preferentially selecting for this type of generalist resistance? So the first thing we looked at, sorry if the colours don't come out too clearly, the first thing we looked at is just let's continuously use the ACCA's herbicide. And what we found was that what the models predicted was that you got evolution of this target site resistance after 4 or 5 years, and that was borne out by observation in the field. And you got some, because this target site resistance gives higher levels of resistance, it was preferentially selected over the metabolic resistance, although we did get some enrichment for metabolic resistance. We then looked at what happens if you use the ALS herbicide once ACCA's resistance has evolved. So this is the sequence strategy, typically. And we found that we didn't select for target site resistance to ALS because we'd enriched this metabolic resistance, which gave resistance to ALS. So what we've found then is after another 2 to 3 years, 3 or 4 years perhaps, we saw evolution of metabolic resistance to the ALS herbicides. We then did a simulation where we rotated between ACCAs and ALS. And what we found there, what our models predicted, was that under that scenario you wouldn't select, or the predominant selection would be for the metabolic resistance. But interestingly, what we found was that this mechanism results in failure of both of those modes of action, and we saw that mechanism of resistance evolving after sort of 6 or 7 years, whereas under this strategy we saw resistance to both modes of action after 8 to 10 years. So this was this led us to question whether the sort of much promoted strategy of herbicide rotation may actually preferentially select for the type of resistance that we really we're very keen to avoid. So much of my research and much of what we've got going on now, which I'll come on to talk about, is really motivated by better understanding this non-target site generalist type of resistance, because I think it really is the major problem. So there was another sort of, at the time I was working in Australia, there was another contentious debate around the idea of the impact of low herbicide rates on evolution of resistance. As I said before, low rates were frequently used in Australia because of economic reasons. Australia had a major resistance problem and seemed to have come to that problem sooner than many other parts of the world. So there was a question as to whether that was due to this widespread use of low rates. And there's been quite a contentious argument about whether low or high herbicide rates most rapidly select for resistance. And you can sort of spin it both ways. And the truth is, I still don't think we fully know the answer. But anyway, Johnny Gressel put the argument in the sense of a catch-22, where really the high rates would select for the target site type resistance and the low rates would select for the quantitative type of resistance. And you could argue that if we could preferentially select for one over the other, we would want to select for monogenic resistance. Johnny Gressel has come up with and has used models to come up with quite an interesting strategy where herbicide application rates are varied year on year, and you find some middle ground between you vary rates year on year and don't lead. And sort of find a middle ground where you don't rapidly select for either of those. It's an idea. It's never really been empirically tested. So anyway, just to sort of set up a model for how we might think about this question, we know that within weed populations there will be some inherent variability in their sensitivity to herbicides, and this normal distribution just represents that variability in an untreated, with no previous history of exposure, the variability in those populations. So what we might argue is that a high dose selects outside of that range of inherent natural variability, and so the only way to evolve resistance to those high doses is through novel major gene mutations which greatly shift the resistance phenotype. The counter-argument is low herbicide doses which select within that range of standing genetic variation will preferentially select for the quantitative type of resistance. So we set out to test that model using a well-characterized susceptible Lolium rigidum population and using the herbicide, an ACCA's herbicide, diclofop methyl. So this is a dose response for a sensitive population. This is the field application rate. We asked the question, we often see a dose response which looks like this, and that's sort of indicative of there being some variation for herbicide response within those populations. So I just set out to ask the question, okay, well, you know, what is there a genetic basis to this ability to survive at very low rates? So this started out as one of those experiments where you just do a look-see, and actually the result, you know, I might have done things a little bit differently if I'd have envisaged quite how startling the results would be, because we really only started with a very small population. At each of these doses we sprayed 100 individuals, and we got 39 survivors at a tenth of the field recommended rate. Now, no farmers are using rates that low, but this was just sort of an experiment for proof of concept that there was heritable genetic variability at low rates. So we took those survivors and we bulk crossed them. Now, the key thing to remember here is this is 39 individuals. This is a very small genetic base. Everything that follows is based on selection from 39 individuals in a susceptible population. And what we found was quite startling, or startling to me at least, was that with recurrent selection over 3 generations, we were able to increase the resistance status of that population by 50-fold. So I guess some of the work we're still engaged in is really trying to understand the underlying mechanisms and genetic determination of this, but a clear demonstration that there's standing genetic variation within these populations which can be rapidly selected to give very highly resistant populations. Subsequent work has shown, or some work we did back in the early mid-2000s showed that this wasn't due to target site resistance. So this here just confirms, I won't go into details, but that was a confirmation for us that it wasn't due to target site resistance, and subsequent resistance in the subsequent work in the lab in Australia has confirmed that it is due to herbicide metabolism. So what we selected was this sort of metabolic quantitative resistance. So sort of motivated by that observation, we wanted to understand really if this was a general phenomenon. So we were fortunate in being able, as I say, ryegrass is a pasture species in Australia, and there are some areas of beef and cattle production where ryegrass is grown as a pasture species. They're remote from the arable regions, so we can sort of minimise any influence of gene flow from surrounding resistant populations and go and collect ryegrass populations where we can be fairly confident they've had absolutely no history of exposure to herbicides. So that's what we did. We managed to collect 31 populations. We established that there was, we screened 1,000 individuals in each population, and we established a mean survival of about 0.5%. So 1 in 200 individuals which were at least suspected to be resistant within those naive populations. Much higher, typically in our models we would put in initial frequencies of resistance of something like 1 in 10 million individuals. So of course the important question was what we were observing there heritable? It was. We took a subset of 5 of those populations, in each case just comparing the red lines show the dose response of the naive population versus the blue lines which show the dose response of the once selected population, and clearly what we're picking up in those populations is heritable resistance. We also looked at cross-resistance patterns. So from one generation of selection with diclofop-methyl, what were the consequences on resistance to other herbicides? And we found quite diverse cross-resistance patterns. And actually so down each column we're just looking at cross-resistance to within those 5 populations to other herbicides. And what's interesting is those cross-resistance patterns are quite diverse. So again, what it's telling us is that actually probably the genetic basis of resistance in each of those populations is subtly different. So it's complicated. So we've also, moving on to a sort of different area of research, we've had a long interest in fitness costs of evolved herbicide resistance, and this was a paper in New Phytologist where we really set out some principles for conducting these resistance studies, or these costs studies. So in the past, and it surprises me that many of these studies still get published in the weed science literature, where people take a resistant population and a completely different susceptible population from a different location, and they try and infer whether there's a cost of resistance by comparing growth, life history traits within those two populations, which is a completely flawed design, because you've no way of knowing whether any differences you find is due to resistance or just due to other. It's a completely you're comparing resistance and susceptibility in a completely different genetic background. A slightly better approach is to compare multiple R and multiple S populations, but what the approach we've used is to often resistance is not fixed in populations, so the approach we've used is to segregate out resistant and susceptible lines from a single population, and then compare fitness of resistant and susceptible individuals from a single population. So for this particular population, a well-characterised ryegrass population, we know it had susceptibility maintained in the population, and it had metabolic resistance and target site resistance. So we were able to segregate out seed populations for each of those three phenotypes, and then do fitness studies to try and establish whether the target site and the metabolic resistance were associated with fitness costs. An important and sometimes overlooked aspect of these studies is to study fitness costs throughout the life cycle, and this is what the student Martin Villarreal did during his PhD. And Martin found some interesting results. We found that the germination dynamics for the target site resistance were quite different to both the metabolically resistant and the susceptible portion of the population. So deeper seed dormancy and a light requirement for germination. These results have been sort of backed up with other populations subsequently. So this just understanding these differences provides some potential for thinking about how we might design weed management systems to actually take advantage of these Achilles heels, or these costs, or these life history trade-offs which are associated with resistance. What Martin also found was reduced vegetative biomass in the P450 and a reduced relative growth rate, and he found in competition with wheat, that the metabolically resistant population produced 23% less seed. So a fairly substantial fitness cost. Now how we might use that in management is open to question. Finally, what Martin also did to bring this all together were field trials to just look at how, starting off with basically with the population with a mixed population where we had 10% susceptible, 30% ACCAs resistant and 60% P450 resistant. We looked at how the frequencies of those three different mechanisms changed over time in the field. And what we found was a big increase in the number of susceptible individuals and a reduction in both P450 and target site resistance. So these fitness costs were present, and they could lead to quite rapid regression of resistance if selection pressure was removed.